question. Am I muted?
No, we can hear you.
No, we can hear you.
Okay, so I'm going to talk about Klotho. She was named after Zeus' daughter, who spins the thread of life. Can you see my arrow?
Yes.
No.
Yes.
So Zeus had 3 daughters, Klotho, Atropos, and Lachesis. Klotho made the thread, Atropos measured it, and Lachesis cut it at the end, which indicates death. So this is according to the Greek mythology. And the person who named the gene after Klotho, you will see in a moment why he did that. So Klotho was, the gene Klotho was discovered by mistake when Makoto Kurō in Japan wanted to inactivate another gene, but what he obtained was a mouse that showed very strong hallmarks of aging, actually even human aging. The mouse, 50% of the mice are dead by 60 days. And they suffer from, you see the long list of osteoporosis, emphysema, but what was interesting to me was the cognitive impairment, synaptic loss, hippocampal degeneration, also motor neuron degeneration, and premature death. And the conclusion was very clear that one protective gene is responsible for many age-dependent characteristics. And this is a slide from one of his papers. The Klotho knockout, as I mentioned, has an 80% shorter lifespan than normal mice. Here are the normal lifespan of the mouse. On the other hand, Klotho overexpression, the mice live instead of 2 years, 3 years. So there is a 30% increase in lifespan. So having more Klotho, and it's only 1.5 more or 50% more Klotho, not a lot more, can prolong lifespan. And also health span, which is very, very important. So in his first paper, he showed by RNA gels that Klotho is mostly expressed in kidney and in the brain. And this is from the Allen Brain Atlas. You can see in the brain of the mouse, Klotho is very strongly expressed in the choroid plexus, but also in hippocampal neurons, which is very important for us. And we discovered in the brain, and others discovered in other organs, that Klotho levels are downregulated as a function of age. In the same time where we see an increase in neurodegenerative diseases, cardiovascular, kidney diseases, and cancer. In all of these conditions, Klotho plays a very, very important role. But today I will focus, of course, on neurodegeneration. I will not talk about kidney or cancer. So Klotho is a transmembrane protein, very similar to APP. Not only that it's similar to APP, but even alpha, beta, and gamma secretases cleave it. We published that 2007. So Klotho is a transmembrane protein with a very short intracellular domain and 2 extracellular domains, KL1 and KL2, that are very homologous to each other, and also possess activity of an enzyme, a glycosylase. Glycosylase is an enzyme that removes sugar from glycosylated proteins. And by doing that, it can modify the function of some proteins that I will mention. And the transmembrane Klotho is shed mostly by alpha secretase, and also cleaved here also by alpha secretase. And those pieces are released, and they are circulating. We can identify them in blood, in urine, and in the CSF as well. There is another differentially spliced isoform of Klotho, which is identical to KL1, but has a little tail of 15 amino acids, which are very different from human and mouse, which is interesting by itself, thinking about evolution. And our collaborator in Barcelona with the first author, Masso, showed that this form is predominantly expressed in the brain. So Klotho is a very pleiotropic protein. It has many functions. One, as I mentioned, as a glycosidase, removing sialic acid from a glycosylated, for example, TRPV channel. This is in the distal tubular epithelium of the kidney. By doing that, the TRPV channel interacts better with galectin-3, and as a result, the channel resides longer on the membrane, allowing for more calcium entry. Klotho is also inhibiting Wnt signaling because it binds to Wnt, so Wnt cannot bind to its receptor. One of the very important role of Klotho in the kidney, in the distal tubular epithelium as well, is to be a co-receptor for FGF23 signaling. So the FGF receptor itself cannot bind FGF23, which is important for the homeostasis of calcium, phosphate, and vitamin D, without having Klotho next to it in the membrane. And there are other roles of Klotho that I'm not going to in because it will take a long time, but also it inhibits IGF-1, for example. That probably, yeah, it is shown here. This is also a slide from Makoto Kuroda, the discoverer of Klotho, who is a collaborator of ours. And he mostly works on the kidney. And this is a diagram to show that as a transmembrane protein, Klotho is a co-receptor for FGF23 signaling. The secreted one, or the shed one, because it's not really secreted, it is shed, has roles as either inhibitory, or it either inhibits or activates blue and red various ion channels, for example, the potassium channel and some calcium channels, affects phosphate transport in the kidney, and it inhibits IGF-1, Wnt, and TGF-beta-1 signaling. These are all important roles that the circulating Klotho plays. So, but I'm going to talk about the CNS today and the neurodegeneration, and I'm going to show you, this is more or less the outline of my lecture, how Klotho affects neurons, cognition, and Alzheimer's disease, how it affects oligodendrocytes, myelin, and multiple sclerosis, microglia, astrocytes, and neuroinflammation. And as a model, we have amyotrophic lateral sclerosis, ALS. There are other groups that worked on Parkinson's disease, and in 30 minutes, I don't have time to talk about Parkinson's disease, so I will not. I will just mention PTSD. And these are some of our publications, the last one in 2020. So I will start with the neurons. We wanted to know if we add Klotho to neurons, to primary neurons, what happens to them? So can Klotho rescue them from a variety of insults? So as the first insult, we took glutamate, and we isolated primary neurons, hippocampal neurons, from rat or from mouse. And we showed that if we pre-incubate the neurons with Klotho and we add glutamate, there is less death. Here in A, there is also less ROS, reactive oxygen species, that are formed when Klotho was pre-incubated with the cells. We see the same thing in LDH secretion. And if we use the HT22 hippocampal cell line, we see the same results exactly like with primary neurons. And let's go to the next slide. We took another insult to the neurons, which is A beta, and we had two types of neurons. We purified primary neurons from wild-type mice and from overexpressing mice. These mice are Klotho overexpressing. As you can see, we are purifying the oligomeric form of A-beta, which is known to be more toxic. And when we add either exogenously recombinant Klotho or endogenously, which is the Klotho found in the overexpressing mice, we can see that having Klotho, when we add a beta at 1 or 10 micromolar, we see less cell death, less cytotoxicity if Klotho is present. And in this paper that was published a few years ago, we showed that among the genes that were affected by having Klotho was a couple of genes. One is peroxyredoxin and one is thyroidoxin reductase. These are two genes that are involved in oxidative stress, and they take care of free radicals. They get rid of free radicals. So if we use shRNA to peroxyredoxin 2, we can see that we cannot rescue as well those neurons. So we hypothesize that Klotho inhibits glutamate toxicity and A-beta toxicity by binding to a yet unknown receptor on neurons, activating AKT, phosphorylation of FOX, and activation of peroxyredoxin and thyroidoxin reductase that lead to neuroprotection. This was published 6 years ago, actually. So this was all in vitro. Now we move to in vivo in collaboration with Leonard Muki from UCSF. We crossed Klotho overexpressing mice with his APP mice that make plaques, and they have problems in the Morris maze and with LTP. And we see that both in the Morris water maze, red are the APP mice that don't perform well, and it takes them a long distance to find the platform. On the other hand, the doubly transgenic mice, APP with Klotho, they perform as well as control mice. And actually the Klotho mice are better than all of them. And here we see the same with the LTP. The Klotho mice are better than non-transgenic mice. They perform better, their LTP. These are the APP mice, and in the middle, or pretty close to the wild type mice, we can see our doubly transgenic, that we rescued the phenotype that APP and A-beta influenced the mice. So there were some papers recently, and I was asked to comment on them. Klotho has a number of polymorphisms, and one is in the coding region. One is called Klotho VS or KLVS because 2 amino acids are changed into a valine and into serine. So it's called Klotho VS. And these authors discovered that people have to move the pictures a little to the right so I can see. Yes, it jumps back. People who have this KLVS, but they are heterozygous to KLVS, and they have ApoE4, they have less amyloid beta than people who have ApoE4 but don't have KLVS. So KLVS is protective in people who have ApoE4. And after this work, after this work, another paper just came out in JAMA Neurology, and this was the one I was asked to comment for ALS Forum. They took meta-analysis of over 20,000 people. This included MCI controls and Alzheimer's disease. And they found that the carriers, ApoE4 carriers that have KLVS heterozygosity have less Alzheimer's disease risk, which means if they have MCI, they will not develop to AD. they will stay as MCI. And this is shown here. This is people who don't have KLVS, they develop, the risk of conversion is higher than the risk of those who have the KLVS polymorphism. On the other hand, people who don't carry ApoE4, there is no difference in the risk for Alzheimer's disease, for conversion. And in addition to that, also they have higher A-beta in CSF and lower A-beta on PET scans, which means the A-beta is being transferred to the CSF. We measured, when we saw this, the question was, do people that have ApoE4 also have more Klotho in their blood? And indeed, the question was, can we find a difference in the levels of Klotho if people have ApoE4? That was our question. And in ApoE3, both females and males, we didn't see a difference in the levels of Klotho. But in ApoE4, at least one allele of ApoE4, we saw that AD have less than MCI and have the less than control. And this was only true for females and not for males for some reason. But the samples were very small, only 15 patients. So this needs to be confirmed with a larger group of people. So this was all about Alzheimer's disease for now, and we are going to move to oligodendrocytes and MS. We were very, very surprised to see this EM that we prepared of the optic nerve from wild type mice and Klotho deficient mice. In Klotho deficient mice, in optic nerve, in spinal cord, in corpus callosum, only 10% of fibers are myelinated, compared to 90% in a wild type mouse, which means that there is severe demyelination or dysmyelination in mice that don't have Klotho from birth. So we wanted to know if Klotho affects oligodendrocytes. So we cultured primary oligodendrocyte precursor cells, or OPCs, and we treated them with Klotho. And then we measured, we used markers for all oligos, which is OLIG2, or only mature oligos, which is CC1. And we calculated the ratio with and without Klotho, the addition of exogenous recombinant Klotho. And we saw that in two different types of media, 3 days and 6 days and 8 days in culture, with Klotho in black, we see more mature oligodendrocytes, the ratio of mature to immature, than without Klotho. This was published already a long time ago. And another experiment that we did was to add Klotho and do PCR for something that is called OPC, maturation enriched genes. And we found that most of the genes that are found in when OPCs become mature are increased in the presence of Klotho. Then we moved to in vivo experiment, and we used the Cuprison model in which there is severe demyelination after mice are fed for 6 weeks with Cuprison. You can see here, this is EM without Cuprison. We did the experiment in wild type and Klotho overexpressing mice. In the beginning, there is no difference in the number of myelinated axons. After 6 weeks in Cuprison, 95% of axons are gone. Here the numbers are 10,000 and here 50. So the demyelination is very similar in both, but after 3 additional weeks of normal chow, the Klotho overexpressing mice myelinate twice as well as wild type mice, suggesting that having Klotho is important for remyelination. So we hypothesize, and actually we have data to show, to prove our hypothesis, that Klotho action via receptor induces OPC differentiation. We see this, that it is antioxidative from the neuronal experiments. And I will show you that it's also anti-inflammatory, and all this is important for remyelination in the white matter and gray matter as well. So let's move to ALS. I'm seeing my time is going fast. So we took SOD G93A mice and crossed them with Klotho overexpressing mice to generate Klotho SOD biogenic mice. And we analyzed wild type in all 4 groups that were generated for neurologic scores, weight, grip strengths, disease onset, and lifespan. And we show here, we measure how many seconds a mouse can be hanging on these wires. And you can see in pink and green, in pink and green, these are the Klotho mice and the wild type mice. They can stay more than 30, but we don't let them because there is no reason. The ones that are SOD, they fall after 10 seconds. And the ones that are doubly transgenic in blue, both males and females, and it is significant difference that they can stay longer. The grip strength is stronger in the doubly transgenic mice. Also, Klotho overexpression extended survival in these SOD mice. Those are mice that they have a very aggressive form of the disease, but we did prolong their lifespan by maybe 2 weeks. It's not a lot, but if we translate it to human time, it will be something like 6 months. And when we did some RNA-seq, we found that in motor cortex and in the spinal cord, we have genes that are related to inflammation that are downregulated, and genes that are important for antioxidative stress and myelin formation are upregulated in the presence of Klotho. And when we looked by immunohistochemistry in cervical and lumbar spinal cord in these mice, looking, first of all, the most striking results are the IBA or the microglia. You can see the SOD have a lot, Klotho overexpressing and wild type have less, but the doubly transgenic, they have a big decrease in the amount of inflammation in the form of activated microglia. We also see this with GFAP, almost significant. And also we see a loss of the neurons that is completely reversed in the presence of Klotho. So since we see decreased Klotho expression with aging in Alzheimer's and MS, in chronic kidney disease and in cancer that I didn't talk, we decided that it will be a good idea as Klotho levels decrease with age and pathological processes increase with age, and Klotho is involved in all this, to try to increase the levels of Klotho. And in the annual report of ADDF, there was a list of 7 targets in Alzheimer's disease drug discovery that are now in clinical trials. And it looks like Klotho boosting that can cause resilience to insults can work on both inflammation, neuroprotection, synaptic activity, mitochondria, and vascular. I didn't go into all of this. And maybe it can be used in conjunction with beta amyloid and tau therapies if necessary. So usually the treatments today or in clinical trials, they are only targeted to one of these possibilities, but we hope that Klotho can maybe affect many more. So one way that we want to increase the levels of Klotho, and we published this in 2018, was by CRISPR's transcriptional effector complex. And we show, after finding a number of guide RNAs, 1, 2, 3, 4, I show where they are located in the promoter of human Klotho gene. we can see that the guide RNA3 is the best in this cell line HK2, which is a kidney cell line. By PCR, we can see an increase compared to empty vector. And we can see also by Western blot, compared to EGR1, which is a positive control, that we can see an increase with guide RNA 3 and 4. We can see this also in SY5Y cells, and the same here. Another way to increase Klotho is to activate the promoter with zinc finger proteins, and this was just published in January of this year in FASEB, that we found several zinc finger proteins that can activate Klotho expression. And here is an example, the paper is out. And we have two types of such zinc finger proteins that we designed. One were constructed from modular building blocks, and some from naturally engineered EGR1 transcription factor backbone, which is known. So we took pieces and put them together until we got this kind of increase in expression. So I'm down to conclusions that the anti-aging protein Klotho is downregulated with age. Epigenetic changes in the form of promoter methylation may partly explain the age-related and cancer-related downregulation of Klotho. We published this, but didn't have time to talk. Klotho is involved in oligomaturation, and its age-dependent decrease may affect myelin repair in normal aging, in Alzheimer's disease, ALS, and MS. Klotho protects hippocampal neurons from A-beta and glutamate toxicity and lowers oxygen species. Klotho reduces anti-inflammatory cytokines in primary microglia. Klotho overexpression delayed disease onset and progression, mitigated neurological deficits, and extended lifespan in the SOD1 mouse model of ALS. So Klotho is a neuroprotective, antioxidative, anti-inflammatory, and pro-myelinating protein. Thus, increasing its levels would be beneficial in neurodegenerative diseases. And I think I did it exactly in 30 minutes. And these are my colleagues who did the work, mostly Sidi Chen, Ella Zeldic, and Andrea Yuste from Spain. I'm done.